Okay, good afternoon. So, sorry for the delay. There is some network issue. We're not able to connect to the internet. This will be my last lecture. So, what I'm going to do is I'm going to talk about additive manufacturing of brain phantoms, both for brain stimulation as well as for traumatic brain injury and concussions. Okay, so this is just the disclosure, which I have to every time. Whenever you have a company, you need to display that. So again, I talked about brain stimulation a lot. We went over the motivation why we need neural engineering or neuroscience studies related to the brain. We need to understand the brain better, and we also should come up with tools and techniques that can study the brain better or treat the brain better. Okay, so we went over these things a lot. So I'm focused on this one, the transcranial magnetic stimulation technique, where we use magnetic fields on the brain to stimulate neurons, make them fire. By doing that, you can either downregulate or upregulate neurons, and by doing that, you can actually modulate different functions of the body. So that is the main concept behind TMS. So this is surgery free, no side effects reported, and you can administer this treatment in outpatient setting, meaning a nurse can administer this if the doctor prescribes it. You don't need to be hospitalized to get this treatment. And again, I went over the underlying principle, how neurons fire. So Faraday's law of induction, a time-varying magnetic field minus dB by dt will induce a curl of electric field. So there will be electric fields, these circular electric fields induced in a conducting material. And when that crosses a threshold field, the neurons will fire and depolarize the neuronal membrane. That is what is neuronal firing. And once the neurons fire, that information is carried on to the next group of neurons where they're connected, and that changes a certain function of the human body. Okay, so you can make them fire at a higher frequency or lower frequency. That is, you can upregulate them or downregulate them. Okay, though it seems straightforward, but it's very complicated because the induced electric field is a vector quantity. So depending on the position of the coil, location of the coil, so the induced electric field on the neuronal membrane changes with the position and location. And to make things even more complicated, all of our brains have different size, different shape, and different curvature inside. The gyri and the sulcus, they're called the foldings in the brain. They have different type of foldings, right? So each person has different foldings. So that means if you apply a magnetic field on one person to get evoked potential or motor evoked potential, you may not get the same effect on another person with the same magnetic field, right? So that means it's the dose change, the functional state of the brain is also because of the geometry. Okay, so two things matter. How much field is needed to stimulate somebody's brain? That is geometry and the functional connections between the neurons. Okay, so let's first look at the geometry. We can come to functional connection because that's slightly more complicated, right? So the geometry, as I said, if we know the rate of change of magnetic field, we can calculate the induced electric field. And because the geometry is so complex, it's not a simple sphere, so we use finite element analysis to calculate the induced electric field, or vice versa. If we know how much electric field is needed to make the neurons fire, what is the needed magnetic field? So we can do these calculations, and we have done a lot of calculations for designing new coils, right? We wanted to use new materials, new magnetic materials, and design different type of coils to target different areas of the brain. Because different areas of the brain need to be stimulated for treating different disorders. If you want to treat depression, you target left dorsolateral prefrontal cortex, basically emotional part of the brain. If you want to target somebody having schizophrenia with auditory hallucinations, you target primary auditory cortex, right? If you want, feel free to ask questions. What if it's what kind?
Anxiety.
Anxiety. So anxiety is much more complex. Some diseases originate from a certain specific region. Other diseases come as a combination of multiple, with malfunction in multiple areas. So anxiety, again, your prefrontal cortex plays a major role, but there is also the fear role that comes from amygdala. So it's a multiple region disorder. Again, people are trying to identify where you can stimulate to reduce the symptoms, right? So even Parkinson's is the vibrations, right? Tremors. So you think, okay, if I can target motor cortex, you can reduce tremors, but that's not the case. You have to target some other area. So it's different location, different signal strength, and different shape of coil is needed for treating different disorders. So we have designed different coils, design, meaning theoretical studies, right? We have modeled different coils, but how do we test them? We can't just go and put on somebody's head because that would be dangerous, right? Even if you want to do animal studies, it takes a lot of time. You have to get approvals. Getting approvals itself is very time-consuming affair. And also, even if you get the approval, housing or rearing animals and monitoring them is expensive, especially in the US, because there are so many rules, so many procedures we have to, or so many laws we have to follow. So it makes it expensive and time consuming. So if you design a coil, you can't just go and test it on an animal. And even worse for human beings. So although we had a lot of models from Parkinson's patients, from schizophrenia patients, from depressed patients, from TBI patients, we designed coils for different diseases, different type of coils, but we never tested them. So, so what's the solution then? How do we do it? So that's where we need brain phantoms. So now I know how to take your MRIs and then build a head model specific to that person. So once we have head model or phantom, what we call as phantom, that mimics the structure of a certain person. Now we can test on this first, right? And then do the animal studies, right? So but there was nothing like this that existed before. So we had to build this brain phantom ourselves. So we had a lot of computational head models of Parkinson's patients, for TBI, all those. Something like we had 2,000 odd head models, but we never validated our designs. So we looked around, phantoms did not exist, so we started to work on making our own brain phantoms. So how do you do it? I mean, nowadays there are so nice 3D printers. You can print, basically make a shell, 3D print them, put a polymer that goes to all the structures and replicates the gyri and sulci of the brain, right? So we can replicate the structure, but still, this is a silicone material which is non-conductive. It might replicate the brain structure, but what about its conductivity, its magnetic permeability, or for stimulation? But if you are measuring stresses and strains due to different impacts, like when you have an accident or you are playing American football where there is a lot of head concussion, or boxing, to measure stress and strain in the brain, this material doesn't work because that material doesn't resemble the mechanical properties of this silicone material, doesn't represent our brain material. So we have to make a material that matches the viscoelastic properties of the brain, right? So we started working on this, and one PhD student spent 4 years to come up with the entire procedure. Okay? So first you take MRIs of the patient, and then you segment all the regions of the brain. This is skin, skull. Below the skull is cerebrospinal fluid. Below the cerebrospinal fluid is the gray matter, white matter, ventricles. Okay? So these are the major parts of the human brain. So we need to replicate these structures. And so we did that. This is where we built the head model like this, segmenting and building, and then the shell creation. We need to create a shell for each of these. And then shells 3D printing, and then we have to come up with the polymer material that goes into these shells. And once you have the polymer material, then you dissolve the shell to get the structure you like. And then you have to confirm whether the structure you got is right or wrong. So you do a CT scan of that. You can't do an MRI of the silicone brain phantom. Do you know why?
It is non-magnetic.
Sorry?
It is non-magnetic.
Non-magnetic, but our brain is, are you saying our brain is magnetic? So I think you have an answer, but the exact terminology is, so how do you get an image in an MRI? It's magnetic resonance imaging, right? Magnetic resonance. So to get an MRI, do you know what is essential component? Protons. Yeah, unpaired protons. Which one? Hydrogen has unpaired protons. And so you need unpaired proton, which means water molecule. Basically, our MRIs are tuned for looking at the resonance frequency of the proton of hydrogen molecule in water, hydrogen atom in the water molecule. Okay? So you need water content. And if we use silicon and make a silicon brain structure, they don't respond to magnetic field because the unpaired proton doesn't exist in a silicon material, right? So water molecule is important to get an MRI. So that's why we have to do a CT scan and then overlap each other. Like, okay, my CT scan of this phantom resembles that of the MRI, so that means our structure matches. If you want to publish anything, you need to do this. You need to say, okay, whatever you have done, you have to validate it, right? So that's what we had to do. We had to take a separate skin, the CT scan of that, to confirm that the structure matched the original structure. And then, so that's just replicating the structure, but now you want to replicate the conductivity of the brain layers. So the conductivity of skin is not that high, so you can ignore it, just create a PDMS, which is silicone polymer. Skull is similar to ABS plastic, so we use that. CSF, what is the best material to replace CSF? Cerebrospinal fluid. It's a transparent, odorless fluid in your head that protects the brain. Sorry? Saline solution. Yeah, you can, so that means if it's saline, meaning its conductivity is also high. So we basically made a saline solution with the matching electrical conductivity of CSF. So that is CSF, gray matter, has certain electrical conductivity. We need to replicate. So we need to replicate the electric conductivity of this, which is not that hard, because by changing the salt concentration, you can get the electric conductivity matching that of human CSF. Here, this is gray matter. It has electrical conductivity of about, I think, 0.28 Siemens per meter. And this one is something like 0.33 Siemens per meter. Then this is ventricles. Ventricles is nothing but these are special regions in the brain which are also filled with CSF. Okay? So these all the properties of these regions should be replicated. So how do you make electrical conductivity polymer? That also did not exist. So we saw somebody trying to make a polymer for some other applications. You take graphene or carbon nanotubes and put it in the PDMS, basically a silicone polymer, and the more carbon nanotubes you put, the higher conductive the composite becomes. So, and we titrated it. We started adding, this is the percent of multi-volt carbon nanotubes. So as and when we started to add more and more percent of carbon nanotube, the electrical conductivity, this is resistivity. Okay, resistivity drops down, electrical conductivity goes up. So the resistivity started to drop. Okay, so there is, or the conductivity goes up. Okay, so there is a certain point where the conductivity shoots up. Do you know why? It's, by the way, it's called percolation point. Do you know why the conductivity increases slowly and at a certain point it suddenly jumps? So think of carbon nanotubes, right? Carbon nanotubes, if you start adding them, they create path for electrons to conduct, right? That's how the conductivity increases. But now, after a certain point, you have added so many carbon nanotubes that you form a network. Suddenly they're touching each other, right? As soon as that happens, there is a direct path, or there is easy path for electrons to transport the charge. So your conductivity suddenly increases. That is called percolation point. You see here, here and here the conductivity is not that high, but as soon as it gets the continuous path from one point to another point, the conductivity shoots up. And that's how this is 5.3% carbon nanotubes, 8.3%, 14%. You see the network here? It's already high. So 14% is already over here somewhere. Yeah, so here the network is already formed. Okay? So that's fully connected network. But we don't need that much because our conductivity is not 8 Siemens per meter. We need somewhere around here. Right? We have small conductivity. So that means you should be careful. You should know where is the percolation point so that you operate at a pre-percolation area. Okay? So that's how we make the polymer conductive, or we make the material functional. Right? Any questions so far? Now, so when you are making the polymer, you have to have high-speed stirrer, and the speed should not be too high or too low. Too low, it won't disperse well. Too high, what do you think will happen if there is too? Sorry?
Bubbles will come.
Bubbles. Yeah. Well, you can remove bubbles afterwards, but other than bubbles, there is another problem. Bubbles is that's
Regeneration will happen.
regeneration. Heat generation. Yeah, heat generation. Yes. Depending on the viscosity, yes, it is quite viscous that heat generation can. But if you have cooling mechanism, what else? Centrifugal force, and also the centrifugal force, you are contained, right? But since there is large amount of force, what will that do to carbon nanotubes? To break carbon nanotubes, and now you are, instead of long carbon nanotubes, you have smaller ones. So you damage the network. You don't get that network. So you have to have the right speed. Again, trial and error. When you are doing all these new things without literature, that's why it took 4 years for the PhD student to go from a MRI scan to a fully functional brain phantom. Okay, because he had to figure out everything, the percolation point, the speed, all those things. Here is the MRI scan, and then final product looks like that. Once you pour and dissolve, once you dissolve the shell, that's how it looks like. You still need some manual removal of some of the undissolved part of the shell. Okay, so now this is how the skin is made out of PDMS polymer. This is non-conductive, and you have to insert the electrodes where you need to measure the induced electric field or the current. And this is the TMS pulse. When you apply magnetic field, that's how it looks like. It's a pulse like this, okay? And if you put an electrode and put a TMS pulse, that electrode will record that TMS pulse in the brain phantom. If the material is non-conductive, you won't see any pulse, okay? So, and when we measure the current or voltage at different points for different field intensities, we get different voltage which matches with our simulation results. Okay? So that means now we have a phantom that can tell you, if you design a new coil and put it on one part of the head, it can tell you whether some critical area in the brain is getting a dangerous field or not, right? Because you don't want to touch brain stem, right? And also you don't want to have so low field that you're not making neurons fire. So you have to have neurons fire just below the coil, but nowhere else. So you can now measure and validate your simulations. Okay? So that's how this is for testing. You can use it for testing new procedure, meaning different field strengths in different areas, or you can also use it to test different coil designs. Okay? So this is how we made a brain phantom that is a result of additive manufacturing of functional material. Okay? And of course, these are our simulations. I've shown these images already. This is the DBS lead, which is the lead that we have inserted. This is the DBS lead. This one. Okay? Here also. But inside it has four electrodes that touch 4 different parts in the subthalamic nucleus, and each one has its own individual wire. It's coiled around and it comes out like this. Okay? And now if we want to do any other, for example, this is a new procedure that we wanted to see if it is safe or not, right? So first we did the modeling to see if it's safe. The magnetic fields coming from TMS on a DBS, deep brain stimulator lead that is implanted surgically in subthalamic nucleus. So we did the calculations and we predicted some things. So if you take the coil right on top, you get unsafe fields, but if it is on mouth motor area, then it's safe. That's what we predicted from our calculations. Because this is meant for Parkinson's patients who have DBS leads in their brain, but if they have speech impairment, the mouth motor area that is responsible for speech impairment or swallowing is somewhere in the motor cortex. Okay? So if you want to do anything to the motor cortex, so one of the proposal was to use TMS to stimulate the mouth motor area to treat the speech impairment. But if you do that, we needed to know it is safe. It doesn't damage the DBS lead, right? Because that is surgically implanted. So we did the calculations and we also looked at what happens if there is electromagnetic interference. Will there be any temperature rise? Unfortunately, there is not significant temperature change, so it's safe in terms of temperature. But if you use TMS coil right on top, then your fields will be not safe. They will be similar to what DBS current has, so which means it can interfere with the DBS treatment. But if it's on mouth motor area here, then it's safe. So here we are showing this is basically if you put the coil in mouth motor area, you get on the mouth motor area good induced electric field, meaning you can make the neurons fire there, but in other areas you don't, this coil doesn't induce large voltages, so it's safe in other areas. Okay? So that's the conclusion from our theoretical study, modeling work. Now we had to prove that with our brain phantom. That's what we did in this study. Here we basically took the same brain phantom, we inserted the DBS lead in subthalamic nucleus and applied the TMS field in different locations. And we see that if we do it right on top, we get currents that are large enough Okay, so that's what we did the worst case scenario here. But if we do on the mouth motor area here, we don't get any of this. I mean, we will have some current in milliamps, but it will be below 0.5 milliamps or something. But if we do it on right on top, we get up to 3.2 milliamps, which is equivalent to what is needed for DBS. Okay, so this is how these brain phantoms were useful to test the safety of this procedure, and also we also can use it for testing the safety of new TMS coils. Okay? Any questions so far? Okay, so then we went ahead and did the same thing for rats. Okay, so for human head it's very easy, and I mean not easy, but it's decent size. Once you figure out, once you know the procedure, it just needs some skills to dissolve the shells and take it out and place the electrode in the right area. But for rats, it's somewhat challenging. Why do you think rat brain phantom is somewhat challenging? Size. Yeah, it's quite small. And for human brain, there was a procedure how to segment different areas of the brain. But for rats, there was none. There was one company that was selling the brain model for rats, but they encrypted it and we could not use it for anything else. We can only run some simulations on it. So we did not have a procedure how to segment different areas of rat's brain. So a really, really ambitious high school student was volunteering in my lab, and he said, please give me something really challenging. I want to take it on. And he was a very bright student. By then he already knew Python, and he had already participated in what is called as Virginia Junior Science Fair and won awards. So he volunteered in my lab, and I said, look, we have a procedure for how to take human MRI, segment different regions. It needs a map, it needs a machine learning work. We don't have something like that for that. So he spent 1 year and eventually came up with a procedure and segmented and built a brain model for a rat from rat's MRI. And we got a patent out of it. And he went to Princeton University to do his undergraduate in biomedical engineering. Okay, so that's I think I might have his name somewhere in the patent. This is him. Anyway, so this is the procedure he came up with. MRI, this is segmentation algorithm, we used 3D slicer. This was much more complex because of the size. And he had to use an existing map. So that is the template, Sigma template. So anyway, so it's a complex algorithm that you have to go through several steps to get to segment different regions of the rat's brain or head model. And now we built a brain phantom, similarly using carbon nanotubes and PDMS, but here it was, because of the size, it was slightly different procedure, and we had to use injection molding. Does anybody know what is injection molding? We need a large pressure to pump in molten metal into a die that will make a shape. So this is injection molding for a rat's brain model shell. Then, because of the bubbles, because we had to do this at a high speed, even one bubble could make a big change in the small rat's brain, right? So we had to put it in a vacuum and make sure all the bubbles come out. Okay, so this is a vacuum chamber, and we finally, this is not the final brain phantom. This is how it looks like. This is the model, and this is the brain. This is the carbon nanotube brain, carbon nanotube PDMS brain. And then adding or placing electrodes inside the brain phantom is also tricky because of the size. So we had to come up with different templates or guides to place the electrodes exactly where it is needed. So that's, and eventually we had to measure the induced electric field. And so because induced electric field is a vector quantity, so it has x, y, z component, right? So we had to measure x, y, z component. Again, we built our own dipole probes, okay? So, and these are the guides or the templates to place the, to make these dipole probes. And then we have guides to place exactly where it is needed. So it's a very hands-on work. And this is done by PhD student Wesley Lord, the brain phantom itself. But the head model creation is done by Chirayu Moker, who was a high school volunteer in my lab. So this is now accepted for publication in Journal of Neural Engineering. And this is also, we did not have, so when the size becomes too small, the induced electric field has to be, the measurement has to be quite accurate. So for that, the setup was quite complex. We used Harvard Medical. Still working. No, I think it just sleep. I think we need something here. Just impressive Microsoft laptop runs. So if it was hard disk drive, I doubt it. It's a SSD because hard disk drives might fail quickly. Recovery of data is easier, but SSD, if they fail, it's impossible to recover the data. Anyway, we did these measurements in Harvard Medical School in collaboration with one of my collaborators, Padma. Yeah, this is where we are matching our modeled work, simulated results to measured results for validating. And then, yeah, this is the way we had to place these. We had to make these separate guides in order to place it where we want. Otherwise, it would be difficult because we need to know exact location of these dipole probes, right? So, with orientation. So, this is somewhat easy in human brain phantom, but rat brain phantom, it's a little difficult. So, this is the procedure how we make the phantom. That should be fun. I think. No, I haven't do it, right? Other than only in the last class it has to come down. also the pads are worn out. So these pads, the two pads are the pads are missing on that side. So maybe that resulted as well. Okay, so that's the brain phantom work. And now we are ready to use brain phantoms for testing different procedures or testing different parts. Okay? So, any questions here? Okay, now that is for brain stimulation, right? Now, so that's electrical part of the brain, right? Now, let's come to the mechanical part of the brain. So if you want to measure deformation or strain in the brain, what do you need to make sure? So if you want to make a phantom, how do you want to measure? What should be the main feature of this phantom to measure the strain, apart from replicating the structure? How do you, if you want to design a helmet, how do you design it? How do you know whether hit here creates more damage, or hit here creates more damage, or hit here creates more damage to your brain? You can predict, but you have to, okay, let's say prediction as well. How do you predict? You have to calculate, right? You have to run some simulation. How do you run simulations? First, take MRI image of a person, build the head model, now take it in your answers, right? And then apply impact forces and calculate the deformation, right? But you have to assign material properties, don't you? To different parts of the. So, what material properties do you assign? You need to know material properties, first of all, right? So how do you know material properties of different brain? And what material properties do you need? I know there are some of you are mechanical engineering students. Of course, in a simple case, it would be Young's modulus, right? Young's modulus, if you know Young's modulus, you can, if I put 10 newtons of force, I get 50 microns of deformation because my Young's modulus is 60 gigapascals, right? Something like that, right? That's what the software packages calculate in a very complex shape. But is there this one Young's modulus? Will Young's modulus give you the right result? No, right? So what properties do you need to assign? Because this is not just linear elastic material, right? It's non-linear viscoelastic material, right? So you need what is called as G prime, G double prime. This is a complex modulus. G prime is storage modulus, G double prime is loss modulus. Right? These two make up one modulus, which is like Young's modulus, but for non-linear materials that are viscoelastic in nature. Right? So viscoelastic means there is elasticity to the material and there is viscosity to the material. Okay? So what's the best example of a viscoelastic material?
Rubber.
Sorry? Rubber, what is the viscosity there? Is it viscous? No, right? It's elastic, it's a small load gives large deformation, very high, low elastic. What about, so what's an example of a viscoelastic material? needs elasticity as well as viscosity.
Slime.
Slime, yeah, slime. Or cheese also, slime is more viscous than elastic. So cheese or paneer kind of material is both viscous as well as elastic. Okay? So our brain is much thinner than cheese or the paneer kind of material. So it slides, it is viscous as well as elastic. Okay? So now you have to calculate the strain in the material. So it can only take certain amount of deformation. If the deformation go beyond 3 to 4%, there will be damage to the membrane of the neurons. Our neurons can tear. So that's what you don't want to reach. So that means your deformation for this viscoelastic material should not cross 3%. Right? If you cross 3%, you may not see it in the MRI scan, but you might have small tears in the membranes of the neurons. Neurons themselves are microns, and if you have tears in micron level membranes, is even difficult to know, image in MRI or CT scan. It doesn't matter. You take the best of best scanner or imaging modality, you can't see it. So that's why when people play contact sport, which is like American football or boxing, they get traumatic brain injury and they won't notice that immediately. It comes later on, and if it is accumulated, you know Muhammad Ali, right? Boxer. He suffered from both Alzheimer's, Parkinson's, and whatnot because he had so many concussions. So that means it is important that we analyze what kind of impacts create more than 3% strain in our brain for this viscoelastic material, right? And that's what we are trying to do. There is modeling work. A lot of people have modeled, but again, one thing that is lacking is individualized brain. We have done a lot of brain modeling. We have taken a lot of MRIs for Parkinson's and depression patients, schizophrenia patients, and built their head models, right? Now we can run modeling work and see what kind of impacts create 3% or more strain, right? So that's, which we started this work somewhat recently. Recently means 2-3 years ago. So, but it's recent for these kind of work, okay? So, just to tell you how bad is this. Severe concussions are leading cause of death and disability in people under age 45. Okay? So, there are about 2.5 million emergency visits per year in US related to brain concussion. And 200,000, 2 lakh people are hospitalized and 50,000 deaths per year, 50,000 deaths per year because of brain damage due to concussions. So that's because improper headgear or not good knowledge about where the brain damage occurs, what is the strain level in the brain, what kind of impacts cause. We don't know, right? If we knew, we would avoid certain type of impact. But although our elders might say, oh, don't take blow to the temple, this is the temple of the head, right? Because this is softer area. We don't know that. I mean, their experience might be anecdotal, right? So, we need to do the proper calculations and see all the simulations and predict where the damage is greatest, which type of impact in which area is the highest. So that's one. A lot of people have worked on it already, but again, what lacked was when we saw individualized brain model. They just took one brain model, everybody. One brain model existed of somebody, and then they did it, all calculations on one person. But we know the brain shape and size changes significantly from person to person. So we need individualized analysis for knowing where the damage occurs for different impacts. Okay? So there is a lot of need in terms of health as well as in terms of sports. Okay? So this is a computational model. So this is greater than 4% strain for certain kind of impact, and that causes some tear or damage to the membrane. So there are some attempts to make brain phantom from these head models, but they're not accurate enough. Okay? So we thought, we can do this because we know how to build brain phantoms, and we are in mechanical and nuclear engineering, so we know how to calculate and measure strains in the brain, and we understand the scholastic nature of the material. So, which material to choose? What material matches the viscoelastic properties of the brain? So there are some people use gelatin, but its viscoelasticity doesn't match that of the brain. Some people try phytogel, again, not so, it doesn't match both G prime and G double prime. PVA, same problem. But if you use a composite of PVA, PHY, and some calcium salt, you get what is called as copolymer. So cross-linking happens. So, and you can control this cross-linking by controlling the salt concentration. So you can match the stiffness or the elastic constant of this material. And you can see here, this is an XRD for this hydrogel material. And as you increase the salt concentration, the cross-linking up is more. So when the cross-linking is more, it's acting more like a crystalline material. So this is, as you increase the salt concentration, the peaks are becoming larger and larger. Okay? So, and also the other, if you want to maintain this cross-linking between phytogel and PVA or PHY, so PHY and PVA, you need to freeze it and thaw it, meaning go to minus 17 degrees centigrade and bring it to 20 degrees centigrade. By doing that, you are building cross-linking. So that is very laborious work. Students who do this have to be really dedicated because you have to do it for 12 hours freezing, 12 hours thawing, and you have to do it for 20 times in order to get the exact viscoelastic properties of the brain with these hydrogels. We actually did that. One master student thesis was about that, creating a material whose viscoelastic properties is similar to that of the brain. So now, how do you know whether the viscoelastic properties, how do you measure viscoelastic properties? How do you measure elastic properties? So, we have been saying we have to replicate this, but how do you know we have the same viscoelastic property? You can't go into UTM, universal testing machine, right? Meant for metals. Rheometer, yeah, exactly. We use rheometer. Rheometer will give you G prime and G double prime. So that's a rheometer. So we use rheometer, but for rheometer it takes certain size samples. We have to create these samples. Basically, shears and measures the elastic constant. It has different shear rates, and this is how it gives you a graph. Okay, this is your G prime, this is your G double prime, and this matches that of the brain. Okay, now how do you know this matches that of the brain? From literature. Somebody has already published. You know what they have published, what they have done? That means they have taken actual brain tissue from a human, from a cadaver, somebody's, and put it in there and measured and published it. That is so useful for us, right? Somebody has done the hard work, which is not easy to. If we have to use any tissue biomaterial, we have to change the whole lab setting to become a biosafety lab. Everybody has to have a certain amount of training. We have to maintain all sort of procedures to bring any biomaterial in our labs, which is very difficult. So if somebody already has done it, so we know the proper material properties, and for the composition that matches this will be our brain hydrogel. Okay? So now, is there any other way to measure viscoelastic property? Sorry? DNA. I'm sorry, I'm not familiar with that. Dynamic mechanical analyzer. I'm not familiar with it. Again, I'm not a person from viscoelastic. We just got into the. So DNA apparently is another one. Which does it give both G and G prime and G double prime? Okay, gives constant K, just stiffness K. Yeah, a lot of them give one scalar parameter, but those ones are contact ones. Can you think of any other ones that might be more accurate? Actually, MRI machines can do that. So that is what is called as MRE, magnetic resonance elastography. So magnetic resonance elastography is a new technique. Not every hospital has it. Some hospitals have it for looking at the stiffness of the liver. If you have liver cirrhosis, or fatty liver they call it. If you have liver cirrhosis, the elastic constant of the liver changes. That can be actually measured in an MRI. So without contacting like this, applying a shear stress and calculating the shear modulus or loss tangent, you can actually, without contact, you can use a sonograph or an ultrasound transducer in an MRI to get the elastic constant of the material. Okay, so that's a new technique, but it exists. It can be done for liver, and it's shape dependent. So we basically made a standard sample without mimicking liver or brain, so wherein we can get stiffness constant for MRI, from MRI. Okay, this is what we did, and somehow they were not. We got elastic constant here. If you take the modulus of G prime and G double prime, you should get elastic constant K. This is G prime and G double prime, and K is basically square root of G square plus G double square. Okay? So our K did not match the K calculated from this. That was because we went from a small sample like this to a big sample like this. So we revised that. Now we have a matching measurement, so our MRE measurements match the rheometer measurements. Okay, so now this is a viscoelastic grain. Okay, so it's not black like in before because that one had carbon nanotubes, that's why it became black. This one is translucent actually. It looks white, but if you take it and look wherever there is small thin area, you will see it's translucent. So it's very soft. And inside this, we put a flexible accelerometer, so that will give us the deformation. There. And you see these wires, I don't know if you can see, these are micro wires coming out of this accelerometer. Okay? So now we put this in the brain phantom like this, and here you see all these wires are coming out. These are nothing but accelerometer wires. You connect it to an Arduino, and you can easily measure output from an accelerometer that is embedded in this brain phantom. Okay? This is a known spring constant. We just take it to the back and leave it. Just goes and wax it at a second. And now we have done other work as well. That's the prelim. That's how we started. Okay? And we did the same thing for brain phantom. So here the viscoelastic material is small, and we embedded not accelerometer in this, we embedded a PVDF film that can measure the impact forces. So we tried nanoparticles first, piezoelectric nanoparticles, but it did not work. It has to have that network, which was difficult to attain. So we used just a film, which, because film also will interfere, right? You should not have anything inside the brain that interferes with the deformation of the brain. So here is how a PVDF film looks like inside. We go from here. Don't know whether you see it. And that's my graduate student Wesley. Our university, covered. They published it on news recently. And this is UTM. See, when you measure it on UTM, the strain rate and the force, first, before the skull cracks, this is what you get. Once the skull cracks, this is what you get. Two different type of, and this is from a drop test, ball drop test. Okay, here is the brain phantom connected to an oscilloscope which measures voltage, and we convert that voltage into strain rate by using D33 and G33 constant of piezoelectric film. Okay? And this is ongoing. So we have a rocket club. Students build these huge rockets and go to New Mexico desert and launch their rockets. They participate in competitions. There is in the US rocket launching competitions by different universities. And this is our RAM is a VCU mascot. So this is RAM Rocketry Club. So they were looking for a load. Usually it's satellites, right? But we convinced them that they have to carry our brain phantom so that we can measure the high acceleration, the high G forces. And we put our brain phantom here, and the first was a disaster. The first one broke and leaked all the fluid and the hydrogel. It's very hot area here. So all the hydrogel liquefied and it stank. The smell was really bad, and they said, "We will not carry your brain matter anymore again." It took another 1 year to convince the next set of students, although we had to involve the head of the department to make them carry our brain phantoms. Now they are scheduled, there's one scheduled in March, another launch. So brain phantom will go in there and we'll measure. The first one, well, this is the measurement. So this is when it launches, cruises for a while, and then it fires another second level engine. So these are, every time it fires, it gets large strain. Okay? That's, this is also, we are also planning to put our brain phantoms in a fighter aircraft to measure the same thing. So that is, again, not many people have done this kind of work. So it's somewhat exciting when I talk about this. So that's, this is all made, I mean, this is possible only because of additive manufacturing, right? Without additive manufacturing, we could never replicate the complex structure in the first place, right? So that's, I think that's the end of brain phantoms. I think, so I have to thank my sponsors, National Science Foundation, NASA funded my magnetocaloric work. This is Veteran Affairs, which is the DoD, Department of Defense of US. This is the state of Virginia. They all funded my research. And these are all my students and postdocs over the years. So that's, yeah, that's it. So now I'll take some questions, if you have any. I know you are all like busy with project, presentation, whatnot, because tomorrow is the last day. So I'm happy to answer some questions. I'm here until 4 or 3:45. Yeah.
In the case of Alzheimer's, that is like, you know, on one hand we got Alzheimer's disease, and the other hand was also said, it states that periodic exercise and, you know, reading books, even if they're affected with Alzheimer's, the manner is going to reflect really as well. So, I mean, when Alzheimer's people forget to walk, they become more like a question.
Case by this nun in a monastery, I think Italy or somewhere. No one knew she had Alzheimer's, but all the nuns had signed up to donate their brain for medical study. When they analyzed her brain, it was full of that plaque, the amyloid protein. But until she died, she behaved perfectly fine. Because apparently she was very active, she was knitting, she was doing all sorts of activities. So it did not affect her as much as it affected her. So there is genetic factor to it as well. It's normally these neurodegenerative diseases, they don't come or they don't express due to one factor. They're a combination of multiple factors. Okay, I'm not a doctor, but this is the background really. Yeah, so Alzheimer's is an interesting disorder. Lots of, I mean, it's the hot topic now. A lot of people are trying to come up with how to remove the plaque, or how to reduce the plaque formation. I'm also collaborating with people. We are trying to see how this protein, we are looking at molecular level studies, how to slow down on protein misfolding, why the protein misfolds. And when the protein misfolds in the brain, that's what leads to all this plaque formation. And also, a lot of people have the plaque formation, but then they're filtered out during what is called as G-lymphatic cleaning. The CSF cleans all these neurotoxins in the brain during deep sleep. That's why they say sleep is very important, deep sleep especially, because that's when the CSF filters out. The way, why CSF triggers only during deep sleep? Can we trigger it before, during light sleep or during even awake cycle? Those are the things that are valid questions to see how to remove these plaques. Right? So it's a very much an electrochemical organ. So there is chemistry, there is electricity, you can also use mechanical techniques. So yeah, so it's a very exciting field to be in. And my students absolutely know what they do. Okay, so any other questions? I think that was the next question. I will, yeah, sure. Yeah.
So the question is actually the MRI images are not very much accurate. So did you do any error analysis after modeling and comparing the victims?
So we did the CT scan of the brain implanted, and then we have the MRI. We compare. So that is to show that our structural information matches. Right? So that's. There was one slide where we go back. Nowadays there are a lot of databases. You can just, their researchers are happy to give their data to you so that you can analyze the way you want. You publish your findings. So, yeah, there is a lot of encouragement because there is so much to explore, so much to understand, so many questions to answer. I did not even touch brain-computer interface. Right? So imagine now you have all this information, you connect it to a robot. Neuralink. So they have, Neuralink has a nice brain phantom, by the way. We only recently found out. They only revealed it recently. But their brain phantom is, they are focusing heavy on the cortex, then inside, whereas we look at it. And Neuralink just records the signal. It doesn't stimulate. Okay? If you record and you analyze, decode the signal, that's a complex thing. But once you decode, whether you want to say blue head or that's green, that's all this can be decoded if you record all the neurons. But you know, it's multiple layers of neurons. Each small 1 centimeter cube of the brain has several layers of neurons and they're so fully packed. They all fire. And if you record something here, it's a combination of so many neurons. So decoding that is not easy. Okay. So it's an exciting field. It's not just a biofield. Don't think that. A lot of analytical engineering, a lot of computational engineering, a lot of mechanical as well. So there is so much to explore. I think with that, I'll stop. If you have any questions, I'm around. I can answer your question.